Hi, my name is Drew Harner, and I'm a PhD student at Penn State University where I study viticulture with Michaela Centenari. Here in Pennsylvania, the wine grape industry is spread out throughout the state, and through my projects, I have been able to engage with partnerships with wine grape growers and wineries across the state while also exploring how Pennsylvania climate affects wine grape quality. And this has been mainly through a project investigating regional characteristics of PA-grown Gruner Veltliner grapes and wines, as this grape variety is grown across the state and including at the collaborating vineyards shown on this map. In using these collaborating vineyards, we hope to explore how regional weather conditions shape and affect the chemical and sensory characteristics of Gruner Veltliner grapes and wine. Another main focus of my research is on the spotted lanternfly, the insect shown here. The spotted lanternfly is a plant hopper introduced in 2014 to Pennsylvania, and since then it has expanded its populations throughout PA and into neighboring states in the Northeast and Mid-Atlantic. This invasive insect can feed on over 70 species of plants, but it seems to favor grapevines as hosts and is a serious threat to the wine grape industry here in Pennsylvania and elsewhere. Since it is so new, we are still learning about how infestation can affect grape and wine quality, grapevine physiology, and vineyard health. To explore this, my colleagues and I set up an experiment where we caged Riesling vines and exposed them to different numbers of spotted lanternflies last year in 2019. Last year's experiment helped shed some light on how these damaging insects can affect grapevine physiology and health, and this year we are replicating the experiment to validate these results and better understand how these insects affect the vines. And through this project and my other research, I hope to contribute to the body of viticultural knowledge and assist the wine grape industry here in PA and elsewhere with production of quality grapes and wines. Finally, I'd like to thank all my collaborators, colleagues, and funding sources that have supported my research and education thus far. And specifically, I'd like to thank Banffy Wines. Without you and your support, this scholarship opportunity would not be possible, and I'm deeply grateful for both your support and this opportunity.
Hi, my name is Andréane Baraché. I'm a fifth-year PhD candidate at Brock University. I study biological science with a specialization in plant science, enology, and viticulture. I'm one of the AWSCF scholarship recipients for this year, and I would like to thank everybody at AWSCF, and particularly the funder of the award, the Carroll County Marilyn G. Hamilton Mowbray Memorial Chapter in memory of Peter Aslauer. My research project is in partnership with the Cool Climate Enology and Viticulture Institute at Brock University and is located in the Niagara Peninsula in Ontario, Canada. In my research, what I'm really interested in is to improve the cold hardiness of grapevine by providing permanent solution for grape growers. The way that I'm planning to do this is by finding new clones or rootstocks that could improve the cold hardiness of the grapevines that we already cultivate in Ontario. In the first few years of my research project, I've already demonstrated that clone selection and rootstock selection can contribute to improving the cold hardiness of grapevine. This would allow grape growers to better select plant material so that they don't rely as heavily on Band-Aid solution to protect their vineyard against the cold weather. That would result in a reduction of the economic losses related to the cold damage that they can suffer every winter. There are two reasons why I'm so interested in working in the wine industry and so passionate about it. The first one is that we get to work in close partnership with grape growers of the region. This is very stimulating because we get to help them solve problems that they have, and we get to see the impact of the discovery we make on their crop and on the industry as a whole. The other reason that I'm so passionate by research in this field is that we get to work with specialists and academics of many disciplines, and it leads to very interesting discovery and new approaches to our work. For example, we get to consult with soil scientists or with climate specialists or plant biochemists, and using the different knowledge and tools that they have, we get to improve crops once again and contribute to a better wine industry in general and a more sustainable industry. Once again, thanks to AWSCF, it was an honor to be awarded this scholarship.
British Columbia is one of the premier wine growing regions throughout the world. Part of my research at the University of British Columbia at the Okanagan campus is to look at how some of the indigenous strains found in vineyards locally here can produce more unique wine flavors. Part of my research was taking some of these indigenous strains that we found in the vineyard and fermenting blank Chardonnay juice with them and analyzing the chemical composition of the resulting wine to see how they metabolically differ from more commercial yeast strains. Another aspect of my research involves looking at how smoke exposure, as you can see plenty of smoke exposure in the Okanagan, affects the microbial composition in vineyards. How we do this is we expose grapes to simulated forest fire smoke in isolated tents in a large exposure volume setup. And then we analyze the fungal communities on those grapes using next generation DNA sequencing techniques that allows us to isolate genetic information directly from the environment as opposed to a culture-based technique in which you would have to bring the yeast back and grow it in a lab.
Hi, I'm Laís Moreira. I'm a PhD candidate at the University of Minnesota, and I'm advised by Dr. Matthew Clark. The Minnesota grape industry is relatively new compared with other traditional wine regions in the West. In Minnesota, we cannot grow vitis vinifera, and we rely 100% on hybrid grapes. The hybrid grapes that we grow in Minnesota, they are made from different species, especially vitis riparia. Vitis riparia brings a lot of the good traits, for example, cold tolerance and disease resistance. However, the vitis riparia introduces the grassy and not pleasant flavor to the offspring. To characterize the inheritance of flavor and aroma profile in cold hard grapes, I'm using a map family of 160 individuals where I'm using three different approaches. The first one is the sensory panel, where panelists taste grapes from seedlings for the population that I'm using. And the second step of my research is to conduct a metabolomic analysis. So for the metabolomic analysis, I'm using gas chromatography and mass spectrum to identify and quantify the metabolites present in each sample. So the last step of my research is to gather the data from the sensory panel, from the metabolomic analysis, and from the genomic analysis to make association between those data to understand the inheritance of flavor and aroma compounds in a map family population. I hope the results of my research can help to develop hybrid grapes with high flavor profile. So, and finally, I would like to thank you to Dayton Springfield 08 AWS chapter in memory of Denise Hall, David Pritchard, and Catherine Roloff to support my education and my research. And another special thank you to the Wine Foundation.
Hi, I'm Kim Fang. I'm a PhD student from Dr. Elizabeth Tomasino's research group. I would like to thank the American Wine Society Educational Foundation and the North Alabama chapter for supporting my study at Oregon State University. My research focuses on wine lipids and chemical components that affect wine mouthfeel perception. Let's imagine that you have a glass of red wine in your hand. What would you like to pair it with? Do you think of cheese, steak, or something fatty? They are all good choices because research has shown that drinking wine and eating fatty food help balance the palate and give a unique mouthfeel that could not be found in having only wine or only fatty food. Study on the interactions between phenolics and lipids in model solutions show strong interactions between the two compounds. Therefore, we think that in a complex matrix such as wine, the interaction between phenolics and lipids may result in noticeable impact on wine mouthfeel. Furthermore, our current project studying wine lipidomic data shows a strong application in identifying the uniqueness of wines from different geographical locations. Being a student in this fascinating wine world, I enjoy every single glass of wine using all five senses and thinking about the wine science. I hope that I can learn as much as I can about wine and inspire others to appreciate wine not only as a beverage, but also as an art and a science.
Hi, I'm Clarissa Reyes, and I'm a master's student in horticulture and agronomy at UC Davis, where I am studying drought tolerance in the root systems of native Vitis species. Here at UC Davis, we have a germplasm of grapevines with diverse genetics that have been collected from throughout the U.S. across climates of varying temperature and precipitation. As we experience climate change that drives increasingly arid conditions in the wine-growing regions of the U.S., I am screening several species to see if they have root characteristics that may be advantageous for growing grapevines in water-limited conditions. Within this germplasm, you can easily observe canopy differences amongst individual genotypes, but differences also exist below ground. My research aims to characterize how roots of some of these native grapevines might be able to survive drought conditions. Here, I have constructed low-cost chambers with germination blotting paper as a means to observe differences in root architecture. By identifying anatomical and physiological traits in roots that affect water utilization and transport, we can work towards developing rootstocks that will require less irrigation but maintain current production yields. I am also able to perform hydraulic measurements on these root systems and pair these observations with grapevines grown in more conventional media. I'd like to thank the AWS and the AWS Tucson chapter for their support as I earn my master's. I aspire to serve as a knowledgeable resource in the future to both growers and public consumers as we adapt to the effects of climate change on society and agriculture.
Hello, my name is Yeva Zig. I'm a master's student at the University of British Columbia. The UBC Okanagan campus is in the middle of one of Canada's main wine-producing regions. The sunny Okanagan Valley in the Rocky Mountains is surrounded by forests, and by forest fires. Now, the fires don't burn the vineyards themselves very often, thankfully, but they cause a different threat: exposure to smoke. Smoked grapes result in wine that has been described as having the aroma of an old ash tree, and was not very well received with consumers. The sneaky part of this matter is that the smoke compounds in the grape are hidden. You can neither taste them nor smell them, but they become perceptible in the wine. Research has proposed the following biochemical pathway. Smoke contains classes of compounds called phenols and terpenes. Analysis of grapes has shown that at least some of the smoke phenols can be found in the grape, but in a modified form. Grape cells enzymatically attach various sugars onto the phenolic molecules, making them tasteless and odorless. During fermentation, the sugars get removed, and surprise, you get tainted wine. Our lab is developing analytical chemistry methods to identify and quantify the smoke compounds while still in the berry. We intentionally expose some grapes to smoke, analyze them using liquid and gas chromatography, and compare the results to clean, untainted juice and wine in order to find the best smoke markers. Forest fires cause a lot of damage around the world, but with reliable methods to detect the taint in grapes, winemakers will at least be able to reduce the risk and losses. Moreover, we will be able to develop and test possible preventative measures how to reduce the effect of smoke taint. Thank you very much for supporting my research.